Hi, Kadeem. It's a pleasure to have you here. You're Kadeem Gilbert, so you're assistant professor at MSU, Michigan State University, and you're mostly interested in understanding the interaction between plants and other organisms, which is, in the way you are tackling the problem, not super common, right? So how did you end up studying plants? How did you decide to be a biologist? What's been your path here towards interaction between plants and other organisms?
Yes, certainly. So it in some ways is a pretty tortuous path, as in not very direct, but I've always been interested in science and biology since I was a very young kid. So I decided I wanted to be a wildlife biologist in fifth grade.
Already?
Yes, but I actually wasn't a very outdoorsy kid. I liked reading a lot, and then I liked reading National Geographic and thinking about these exotic ecosystems and that sort of thing. But when I went into college, what I had in mind for biology was really just mammals or birds, these big charismatic organisms, because that's what I would see in books and featured on television. But I took a botany course my freshman year because I figured I'm interested in ecology, so I should know something about plants. But it wasn't until I took that course that I learned, wait, plants are actually really cool on their own right, not just as food for animals. But throughout undergrad, I took as many courses on different taxonomic groups as I could. You know, just not just plants, but also fungi, herps. You know, like reptiles and amphibians. All of these different courses, and I did lots of different types of research experiences in order to. The goal was to better define my interest, but I did very disconnected things like looking at stable isotope labeled arthropods, to look at soil community fluxes, looking at moose populations in New Hampshire, starfish phylogeography in Indonesia, black bears in upstate New York, just very different projects. But every time I did something different, you know, I like the goal was to figure out what I'm going to do for grad school, because I had that as a plan already. I'm going to go straight into grad school, but each time was, oh wait, this is cool too. This is cool too. This is cool too. I actually, by the time that I was finishing up my junior year and it was time to apply to grad programs, I was actually less certain about what I would be doing, but it wasn't actually until I got into a grad program that I realized the answer here. But I mostly applied to herpetology labs for grad school, actually. Mostly herpetology labs, and then a couple of forest ecologists. But at the lab that I interviewed at Harvard, where I ended up going, the herpetologist there could tell that I was not really settled, said, oh, you can do rotations if you come here. But it was through getting to that program and then having the chance to meet Nomi Pierce, who studies symbiosis, and then in her lab, people are studying lots of different organisms. There's at least an insect involved somewhere, but I realized pretty much immediately, it was always the case for me, I guess. You know, I'm interested in all taxa, but I'm most interested in these kinds of interactions.
Yeah.
So
is that how you? So because I mentioned that your way of tackling the interaction between plants and other organisms is a bit unusual, and what's a lot unusual is the organism you're studying and the kind of plants you're studying. Is that, and we go down there soon, I think, but is that where you've started connections with carnivorous plants and looking at those, how do you call them again?
Pitcher plants?
Pitcher plants?
Yeah, so actually symbiosis for me came first, and then the pitcher plants.
Yeah.
But there was a student in my PhD advisor's lab that was already working in pitcher plants, And one thing I didn't mention is that I didn't have a set organism preference defined at the start of grad school, but I had a specific interest in Southeast Asia as a region. I was interested in the ecology of Southeast Asia.
Why was that?
Well,
in a nutshell.
Yeah, in a nutshell, I think for every field ecologist, there's kind of two meccas. There's the Amazon and there's Borneo. And then that's the reason that I decided to take Indonesian language in undergrad. So after also learning the language and having one research experience there, and then learning that there's very few people working there, especially from the West, that it could be a niche that I could fill as someone who knows the language. So that's the other kind of weird connection that brought me to these plants. But like I mentioned, another student was already looking at them. So when I joined the lab, and then I had the chance to to join on an existing project to as a trial to figure out my own interest, I was introduced to this group by this other student, and then learning that they're in Southeast Asia, but also that they interact with everything was the big thing.
Everything meaning the soil, the plant,
so there's
the people.
Inside of these pitchers, there's all sorts of microbes, bacteria, fungi, but then there's specialized insects that live inside of the pitcher fluids, and there's even, in some cases, frogs that are adapted to breed in there, and their tadpoles live in the fluid. So basically, all of these different taxa that I have some interest in, they're in the plant. So
the plant was combining all your previous interests from everything you've been doing over the years.
Exactly. And I didn't think it would be possible to do so, and in fact all of the faculty that I talked to when I was a new grad student were basically saying as their advice, stop trying to find the perfect project that would fit everything. It's not going to happen, but I found something that really worked.
That's really cool. So how did you ended up landing at MSU? What's been your path from there? You've been to Borneo, right?
Yes.
For field studies. Are you still going on field studies, or?
So unfortunately, since 2020, I haven't really done field work. But yeah, so to answer the question about the path to MSU, I finished my PhD and started a postdoc in 2019, end of 2019. So I had a few months of normalcy, and then everything fell apart. But it was quite a roller coaster. On the one hand, thankfully, I had already the project that I was going to be doing anyway was lab based. So thankfully, I was.
You didn't have to travel too much for it.
Right, I didn't have to travel, so it's not like plans got canceled in that sense. But on the other hand, I couldn't even do lab work because Penn State was just shut down for a long time. So it was a 2-year fellowship I was on, and then the first year, I couldn't do anything really. I was able to do some computational stuff that was not really the main plan, but at least partly related to what I was doing. But it was a tough time to be on the job market. And actually, I say roller coaster because there were some really positive things that were happening just before the pandemic, and then I had an experiment that was just ready to go right after spring break, which I was assuming that we were going to come back to, and then that was canceled. But I also had an informal offer from the department that I was in to kind of bridge into faculty from there. But then that kind of went away with hiring freeze. So that's what I mean, roller coaster. So I was freaking out fall 2020, and am I going to get a job? And there were fewer, according to my postdoc advisor, because I haven't really seen a normal year, there were fewer jobs that were available. But at least one of them was this job at KBS, which the Kellogg Biological Station, which sounded wonderful. Living on a field station, someone who's like trained as a field ecologist. So that was one of several positions I applied to. And then I got an interview, which was all on Zoom. That was the world I entered into. But somehow, even through Zoom, I could tell that it seemed like a very lovely department or academic unit. That there's a lot of camaraderie among the faculty and students that are based at the field station. I did at least get to visit before signing. But
and you got there, and then you set up your lab, and so you haven't been on the, how do you call that, in the real environment for 4 years. How do you study pitcher plants and their interaction when you cannot go on site? How do you do that in the lab, and what are the questions you're trying to ask right now in the lab?
Sure. Yeah. Well, as of the past few years, I haven't really been doing anything new with Nepenthes, this tropical pitcher plant specifically. Another complication with the timing of when I started up was slowness of getting some necessary equipment like growth chambers for working with these plants outside of their natural environment. But fortunately, I was already set up to work on different systems because through the postdoc project that I was working on is this comparative study across Nepenthes and a couple of agriculturally relevant plants, a few cotton species and beet. So those were systems I was already working with. So I've been kind of continuing that work, or I guess really started that work kind of around the same time I was interviewing.
So what's the linkage here between these agricultural crops and the pitcher plants?
That's right. It seems like yet another kind of unusual shift that people could look at my path and say, "Wait, how did we get here? How did we get to these crops?" But the connection is, it turns out that so pitcher plants can control pH in their pitchers, which makes sense as they're kind of like little guts. But these are modified leaves. But it turns out that other plants can regulate pH on their leaves too. And cotton is actually quite interesting, being on the other end of the extreme. So for Nepenthes, we can get down to acidic pH 2, and for cotton, we can get up to alkaline pH 10. So these are both extreme plants for pH.
Are both on these variety of plants that you've looked at, are these different pH and regulations of pH impacting the community of organisms that are living either in the pitcher or at the surface of the leaf?
Yes, for sure. So I showed in my PhD that pH regulation in Nepenthes, in the pitcher, very strongly filters microbial communities. And now I'm seeing that the same thing is also true on leaf surfaces, which is ongoing work, which is pretty exciting.
Okay, cool. What are the kind of tools that you're using to study now in your lab on these different agricultural crops? The interaction, I guess, of these bacterial species and the leaf? Yeah.
So the tools in this ongoing work that I mentioned are transcriptomics, so RNA sequencing. So I'm interested both in the host plant, so interested in seeing what genes or molecular pathways can be involved in pH regulation. So
from the plant point of view, or from the bacteria community point of view?
Yeah, so for the plant, so that's from the plant point of view, but also using RNA sequencing of the microbes, which is called metatranscriptomics. So we get the whole transcriptome for all of the microbes on the surface too, to look at gene expression in the context of the interaction with the leaf.
Okay. in this area of science, has there been like a recent discovery either from you or from your colleagues that make you really excited, like on the way plants are interacting with their microbiome at the leaf surface area?
Yeah, well, I guess I just started talking about this new super exciting finding. So just to give a little bit more detail, first is just the finding that leaf surface pH impacts microbiota, but more specifically, I inoculated leaves with a common community, and just after 3 days.
So when you're saying a common community, that means like a synthetic community of bacteria that you've generated?
So not quite, but it's somewhere in between. I started from soil. So it's a wild community in a sense, but diluted it so that we have this complex. It's a complex starting community, but it's from the same source that all plants are starting with, basically the same starting point here. Just after 3 days, the different plant species, which vary in the pH levels they express, they select for a different microbial gene community.
So if you put the same, in other words, so if you take a bunch of bacteria, microbes, let's say, because I guess there are eukaryotes in there too.
Yeah, there could be, though they don't show up in our sequencing data, but they're likely there.
So you take them and you put them in plants with different pH on the surface of their leaves, and that would dictate what's the identity of the bacteria that would live and stay there.
Exactly. And then that you take tubes of that initial inoculum and then leave them out in the greenhouse and in the growth chambers for that same period of time, and then that's kind of a control, and we see that the communities in those don't shift nearly as much as they do on the leaf, and they're not really different.
And what happens if you put them in different pHs? Do they replicate what you've seen on the leaf?
Oh, that's an interesting question. So that's something that I haven't done. You just take that inoculum solution, you don't put it on a leaf, but you add more acid,
you put it on
base. Yeah, that's a good question. I could probably do that easily at a later time.
Okay. And the community, like the scientific community around this, like what's happening at the surface of plants, how do they regulate their own microbial community? Where do you see the scientific knowledge on this going in the next decade or 2 decades? What do you think we'll learn from that?
Yeah, it's an interesting question, because then it's a very small community of people that are looking at microbes on leaf surfaces. A lot more are looking at microbes associated with plant roots.
The roots,
But I could say I anticipate either from my lab and or other labs, either in collaboration or inspired by work going on in my lab. We could, if we can understand, say on the plant side, what genes underpin the ability to regulate pH, that could potentially be a way that we could say genetically engineer crops to express pH levels that stave off pathogens. So it could be a way to have sustainable agricultural production without relying on pesticides, if we better understand how the plants themselves, via this pH regulation, how they control against pathogens and cultivate a healthier microbiome, basically.
And so you think that in the next decade we'll discover the molecular mechanisms involved in this pH regulation? I guess we know a little bit about that in other systems with proton ATPase or stuff like that.
Yeah, it is hard to say because work in my lab is showing that every species might have a different toolkit.
So yeah.
Yes, but if my contention and my purpose with starting with these really extreme plants, the cotton and nepenthes, the idea is that if we at least know how extreme acidification or extreme alkalinization works in this one system, then maybe that could be a trait that we want in tomato or potato or something like that, and say CRISPR it in from one plant species into a new plant species, which is outside of my area, but is an interesting possibility that I think this work could open up.
And just have a quick related question here is like, you're talking a lot about how the plant is affecting its microbiome. What about like the opposite regulations that could occur? What are your thoughts about that?
Yeah, another really interesting question. I talked about my path, how it was kind of long and winding, but I ended up in a place where I am working with everything, but I do see myself as more focused on plants now that I see from the plant perspective, and I have this bias towards thinking that the host has this high level of control. It's likely that symbionts like these microbes, which we know they can also change local pH, there could be interesting negative or positive feedbacks between the plant and the microbes. I might be interested in looking at that at some point, or have a student that's interested in looking at that from the microbe perspective. But yeah, I'll just say that as it stands now, I'm really focused, and I see as kind of a focus of my lab thinking about plant traits and then how that's
the plant size of things,
impacting things.
Makes sense.
Yeah, especially that I know, at least the impression I got as a new grad student is that there's a lot of work from the other side already focused on microbes, and that there's maybe more that we could learn about the role of plant traits in affecting the environment, and then how that affects everything else downstream from that.
And eventually the plant's health, right? That's what you were mentioning.
Right.
Okay, cool. Well, thank you very much, Kadeem, for this nice discussion.
Yeah, thanks for having me.